All right, let's get right into it. For as long as we can remember, the story of Parkinson's disease has always been about one thing: the death of those critical dopamine-producing neurons in the brain. But we're going to look at some incredible new research today that tells a different story. A story about a case of mistaken identity happening right inside the brain's own support crew.
So here's the big question that really frames this whole explainer. What if we'd been looking in the wrong place? What if the real secret isn't with the nerve cells that die, but with the cells whose entire job is to keep them alive? It's a total shift in perspective, looking not at the victim, but at a potential accomplice that's been hiding in plain sight.
Now, of course, at the heart of this mystery is the usual suspect, a protein called alpha-synuclein. We've known for a long time that when this protein messes up, when it misfolds and starts clumping together, it causes big, big trouble in the brain. But here's the thing. What if it's not working alone? What if this protein is actually manipulating an ally and turning it into an enemy?
Okay, so let's meet our new player of interest, the astrocyte. Now, these cells are literally everywhere in the brain. For a long time, we kind of just thought of them as the background crew, but this new research, it puts them front and center, right at the scene of the crime. The question is, are they a friend or a foe?
You see, in a healthy brain, astrocytes are the absolute best neighbors a neuron could ask for. They are the brain's support staff. They do all the housekeeping, they make sure the chemical environment is just right, and most importantly, they help keep neurons healthy and alive. Their whole job is to be protective.
But, and this is a huge but, astrocytes kind of have a split personality. Normally, they're in that helpful neuroprotective state. But when they sense that something is wrong, some kind of danger or stress, they can flip a switch and go into a neurotoxic state. And in this harmful state, they crank up inflammation and can actually start killing cells. So the billion-dollar question is, what makes them flip?
To figure that out, the researchers basically set up a cellular interrogation. They wanted to put our main suspect, the astrocyte, in a room with our main villain, that alpha-synuclein protein, and see exactly what would happen. The experiment was pretty straightforward. First, you grow some perfectly healthy human astrocytes in a lab dish. Step 2, you introduce the bad guy, those clumps of misfolded alpha-synuclein. And then, step 3, you just watch and measure what happens next. And whoa, the reaction was huge. As soon as the alpha-synuclein showed up, the astrocytes just unleashed a firestorm of these inflammatory signals, these cytokines. You can just see on the chart, it's not a small change, it's a massive, massive freakout. They were clearly flipping into that harmful, toxic state. So, okay, the astrocytes are angry. What does that mean for the neurons next door? Well, when the scientists grew neurons in this angry, inflamed environment, things got bad fast. All that stuff the astrocytes were pumping out was directly hurting the neurons, and the damage was devastating. Look at this comparison. When neurons were just on their own, they were fine. But put them with those reactive astrocytes, the rate of neuron death just skyrockets. The very cells that are supposed to be protecting them were now helping to kill them. It's a total betrayal.
And the researchers, they put it pretty bluntly themselves. They said the bad protein basically triggered this inflammatory state, which made the astrocytes stop being helpful and start killing neurons. But believe it or not, the story was about to get even weirder. Because as the scientists started digging into why the astrocytes were so angry, they looked at the genes that were being turned on and off. And they found something completely out of left field. This wasn't just any old inflammation. The cells were ringing a very, very specific alarm bell.
This is the absolute bombshell of the study. The astrocytes were acting like they were under attack from a virus. They were mistaking this misfolded protein for an invading pathogen. It's a complete case of cellular mistaken identity. It's just wild. And the genetic data was crystal clear. The cells fired up all the classic break glass in case of emergency pathways for a viral infection. You see this defense response to virus, response to interferon.
These are the big red buttons the cell pushes when a virus attacks. The protein was triggering a full-on antiviral false alarm.
So when you have this massive false alarm blaring, does the cell have any way to tell itself to, calm down? Well, yes, it does. It's a cool process called RNA editing. You can think of it like the cell has its own internal editor that can go in and tweak the genetic messages, often to try and stop an immune response from going completely haywire. And the main tool for this editing job is an enzyme called ADAR1. It's got two versions. There's the everyday basal version for normal business, but then there's the P150 version. That's the emergency edition. It gets switched on specifically when the cell thinks it's fighting a virus. And sure enough, the researchers saw the astrocytes flipping this exact switch, trying to control the chaos they thought was happening.
Okay, so this is all fascinating stuff in a lab dish, but here's the real test. Is this bizarre drama of mistaken identity and RNA editing actually happening in the brains of people with Parkinson's disease? To find out, the team looked at brain tissue from actual Parkinson's patients.
And here it is, the smoking gun. In the brains of Parkinson's patients, the levels of this specific kind of RNA editing were significantly higher than in healthy brains. The exact same emergency response they saw in the lab was happening in the human disease. This wasn't just a lab curiosity. This was real.
But they had to nail it down. Which cells were actually doing this? They analyzed all the different cell types, and the data pointed a giant blinking arrow at one culprit. Of all the cells in the brain, it was the astrocytes that were overwhelmingly responsible for this increased editing. This was the final piece of confirmation. So let's just walk through this whole tragic cascade of events one more time. It starts with bad alpha-synuclein protein building up. Then, the mistake. Astrocytes see this protein and think it's a virus. Next, the reaction. They launch a massive, but totally misguided, antiviral attack. Then the cell tries to get things under control, ramping up RNA editing. But the final result of all this chaos? It's toxic to the neurons, and they start to die.
So when you boil it all down, the real story here is absolutely incredible. It seems a misfolded protein can actually trick the brain's own bodyguards into a case of friendly fire. They launch this powerful virus-fighting defense that ends up being fatally toxic to the very cells they're supposed to protect. It's a tragic misunderstanding on a cellular level. And this discovery, it just blows the door wide open for the future. It raises this incredibly hopeful question. If we can figure out how to shut off this false alarm, if we could somehow tell the astrocytes, hey, stand down, it's not a virus, could we keep them in that helpful protective state? This could be a completely new way to think about treating Parkinson's, one that could protect the brain like never before.